Welcome to another edition of CCSD Living Environment. Today we're going to continue looking at our introduction to genetics and the work that Mendel did. And to continue looking at Mendel's work, what we need to talk about are Punnett squares. Punnett squares show us the mathematical probability to help predict the genotype and phenotype combinations in a genetic cross. So we need to look at how to actually go about completing a Punnett square. The first thing we need to do is look at both parents. So what I'm going to have is my mom is going to be heterozygous. Remember, that means that they're different. My dad is also going to be heterozygous, meaning that they're different. So what the first thing is I need to do is I need to separate what my parents are, so I know what possibilities the gametes or sperm and egg could have. So for mom, the gametes could have big A or little a, and for dad, the gamete could have big A or little a. So again, what is the gamete for mom? That would be the egg. And what is the gamete for dad? That would be the sperm. So now that I've separated them and I know what the gametes are, what I'm going to do is write each option over one side, and then I'm going to write each option next to a side. Now the thing that we need to do before we actually do the cross is we need to consider what is the gene that we're using. And in this case, the A gene that we're talking about is going to be the gene for plant height. So what does that mean? What is my key? What is that going to give me? If I have big A, it's going to be tall. If I have big A, it would be tall. If I have little a, it would be short. Tall is dominant to short. So all you need to do is go through and bring each box either down or over to fill in the Punnett square. Now, does it matter if I write big A or little a first? It doesn't make any difference. It's just we're more accustomed to seeing the capital letter come first. But it doesn't matter which way you write that. So once you've figured out what options you could get in the offspring, what you need to do is look at that and say, okay, what are the genotypes and phenotypes that I could get? So if I do that with the phenotypes, remember the phenotype is the physical trait. It's what you see. So I would have 3 out of 4 boxes, or 75%, showing me the tall characteristic. And I would have 1 out of 4, or 25%, showing me the short characteristic. That would be my phenotype. That's what I would see. Now let's look at the genotype. With the genotype, I would have 1 out of 4, or 25%, homozygous dominant. Homozygous because they're the same, dominant because they're capital letters. I would have 2 out of 4, or 50%, as heterozygous, hetero because they're different, capital and lowercase. And then finally I'd have 25%, or 1 out of 4, homozygous recessive. Again, homozygous because they're the same, recessive because they're the lowercase letter. And that's the way that you go through to fill out a Punnett square to be able to determine what are the chances of having one type of offspring versus another. How can I get one thing? How can I get the recessive versus how can I get pure dominant? That's what we use Punnett squares to do. So from there, Mendel went through and he came up with this idea of independent assortment. Independent assortment states that genes for different traits can segregate or separate independently during gamete formation. In other words, the information for eye color separates differently than the information for hair color. So just because you have brown hair doesn't mean you're going to have brown eyes. Or just because you have blue eyes doesn't mean you're going to have red hair. So in the case of the peas, just because you're a tall plant doesn't mean you're going to have a purple flower. So what Mendel wanted to know, what Mendel questioned was if one gene could influence the inheritance of another gene. Could having a purple flower influence whether or not you're going to have a tall or short plant? So what he did was he followed these genes for many generations and he ran crosses like this. So now he's running this cross for 2 different traits. In this case, seed color and plant height. So he did it for big R or little r representing the seed color, and big Y or little y representing the plant height. So by doing this with 2 traits, we call that a dihybrid cross. Di meaning 2. Well, what about the trait that we just did? Well, there was only one trait that we were looking at. We call that a monohybrid cross. Because again, what does mono mean? Mono means only one. So when you do it with one trait, it's monohybrid. When you do it with two traits, it's dihybrid. I wouldn't panic. You're not going to have to do a dihybrid cross. Just the monohybrid crosses is all you're going to need to know. So let's summarize Mendel's work. What did we gain from Mendel's work? What were the conclusions, if you will, that Mendel came up with based on what he had done? Well, first of all, the inheritance of biological characteristics is determined by units we call genes, which are passed from parent to offspring. We know that our information, our genetic information, came from our parents. Mom gave us half, Dad gave us the other half. We also know that there are two or more forms or alleles of the gene for every trait, and they could be dominant or recessive. If it's dominant, we represent it with a capital letter. If it's recessive, we represent it with a lowercase letter. In most sexually reproducing organisms, each adult has two copies of every gene. One came from each parent. These genes segregate or separate from each other during gamete formation, so then you're only passing on one to your offspring. And finally, the alleles can segregate, or I should say, they will separate independently. Remember, segregate and separate are the same thing. So those alleles will separate from each other separately, differently. Brown hair, brown eyes. Red hair, blue eyes. Brown hair, blue eyes. It doesn't make any difference. That separation, that segregation is independent. They are not connected to each other. So all of that work that Mendel did led us to some great conclusions and some really important information that broke ground for genetic research. Now remember, Mendel was doing the work that he did back in the mid-1800s. So that was kind of the start to getting people thinking about genetics and how do genes work? How do traits get passed from parent to offspring? So all of that information was very helpful, and as I'm sure you can imagine, like with everything, there's always an exception to the rule. So we need to look at a couple of the exceptions. The first exception that we're going to look at is what we call incomplete dominance. What does it mean if something is incomplete? If something is incomplete, it's not finished, or it's not quite all the way done. With incomplete dominance, the heterozygous phenotype is between the two homozygous phenotypes. In other words, the expression is blended. So if I have a red flower, big R, and I have a white flower, big W, and I cross the two of them, what I would get, the big R, big W, would actually give me a pink flower. It would blend the two of them together. So what would my cross look like? Well, my Punnett square would show in all the offspring of this F1 would all be blended. They would all have pink flowers. Because remember, that's incomplete dominance. One does not completely push the other one out. There is no dominance there, dominance expressed. Because they blend together. The two of them kind of give you this nice blending. This most often happens in flowers, examples being the Japanese 4 o'clock flower and snapdragons. Those are usually the ones where we see that condition. And the next one down that we're going to talk about is called codominance. Well, what does it mean if you have a co-captain or a co-president? It means that both of you are sharing the position. You're both having as much power as the other. One isn't more important than the other. As opposed to a president and vice president, or a captain and an assistant captain, where one has more power than the other one. With codominance, that's not the way it works. The phenotype of both alleles are clearly seen and expressed. The heterozygous is not blended. You see both of them. So the example here happens in cows. So I'm going to use that capital C to say, okay, this is codominant, and then I'm going to have the R to tell me it's red. And then again, the capital C saying it's codominant, and that's going to be my white, the CW. So when I put them together and I have CR, CW, that gives me a condition called roan. Well, what would that look like? If you went up to the red cows and you looked at their hair, you would see all red hair. If you went up to the white cows and you looked at their hair, you would see all white hair. if you went to the roan cows and you looked at their hair, you'd see like red hair, white hair, red hair, white hair, red hair, white hair, because both of them are being equally expressed. One is not dominant over the other. They both share equal power, if you will. So in my F1 cross, both of them show up, red and white, and what they call that is roan. There is an example of this in chickens, where you have one, it's like a bluish hued feather, and then there's one that's just flat black, and so they call that a speckled condition chicken. And then you also see this roan condition in some horses. And again, it means that both of them are being completely expressed. One is not dominant over the other. You see both. And that takes us to the next one, which they refer to as multiple alleles. What does multiple mean? Multiple means many. So I'm going to have many alleles. Well, the genes exist in several different forms. Remember, one individual, one person can only have two of any gene. You get one from mom, you get one from dad. You only have two alleles. That's all you can have. But in the population, there are many different options. So as an individual, you can only have two alleles. One came in from your mom, one came in from your dad. That's all you can have. But in the population, there can be more than two options. And the best example we have of that is human blood typing. We know that we have blood type A, we have blood type B, we have blood type AB, and we have blood type O. So what happens with each one of those? There's a lot of stuff going on here in this key that we've set up to do one of these crosses. Well, let's see if we can figure it out. First of all, I'm using two capital letters here for one condition. That kind of looks like what we did with codominance. Well, if it was codominant, then that would mean that both of them, if they were there, were both expressed. Does that ever happen with blood typing? It does. The AB condition. What does that mean? That means that you've got type A and you've got type B, and they're both being expressed. So that means that you show the condition of AB. What else do we see that's going on here? Well, yeah, I have that capital I to show me codominance, but I also have this lowercase I. When we have capital and lowercase, what is that telling us about? That's telling us about dominant and recessive. So this is telling me then that the O condition is recessive to A and B. If A and B are dominant or codominant, then O is recessive to that. Well, wait a minute, I thought type O blood happened more often in the population. Well, it does. So then how can it be recessive? And that's something that we need to talk about. Dominant and recessive doesn't mean stronger or weaker. It doesn't mean better or worse. It just means if dominant is there, dominant is expressed. Has nothing to do with how often it shows up in the population. So even though this is the recessive allele, it doesn't mean it can't show up very often in the population. In this case, it actually shows up more often in the population. So why are we talking dominant recessive? Because if I have my dominant and the recessive, what do I show? I show type A. Dominant, recessive, I show type B. Both dominant, they're codominant, so I show both. What's the only way that I can fully express the recessive allele? Is if both of my options are the recessive condition. And that's how I end up with blood type O. So when you go through to do a cross for blood typing, the difficulty is in setting up the key. Because not only are we dealing with codominance, but we're also dealing with A and B being dominant over type O. So if we look at a cross, we look at a Punnett square, again I'm going to put my options for my mom, the gamete options, on either the top or the side. It doesn't matter which one goes where. And then I'm going to put the dad on the opposite place. And then all I'm going to do is bring them through just like I did before. And then what does that show me? Here I've got type AB, right, because they're codominant, they both show up. Here I've got type B, because B is dominant over O. Here I've got type A, because A is dominant over O. And then here I will express type O, because then both recessives are there, so the recessive can be expressed. Finally, the last one we're going to look at is this idea behind polygenic inheritance. What does poly mean? Poly means many. What do you think genic means? Gene. So it's a trait that's controlled by more than one gene. So many genes control for one trait. A very good example of that in humans is skin color. There are many, many options to human skin color. It doesn't mean that there's multiple alleles, not in this case. What it means with skin color is that there's many, many genes that are controlling the skin color. Same thing is true with height. There's lots and lots of options for height in humans. And again, what is controlling that? It's being controlled by more than one gene. So because there's more than one gene, we see this wide range of different options. Again, hair color, eye color, many genes control those traits. And the last thing that we're going to look at today is how do genes and the environment combine? What is the effect of the environment on your genes? Environmental conditions can affect gene expression and influence genetically determined traits. We know that our DNA has the instructions. It has the blueprint for what we're going to become, who we're going to be, what we're going to look like, how we're going to express those traits. The individual's actual phenotype is determined by the environment as well as their genes. We know the genes have the plan, but the environment can help to control how that plan is laid out. And that's what happens with identical twins sometimes. If they're not raised in the same environment and one twin is raised in a different environment than the other, the outward expression of their traits can be different. We know that genetically they're the same, but the environment can affect how those genes are actually expressed. And a really good example of that is with hydrangeas, which are a type of plant. If they're grown in acidic soil, the flower is blue. If they're grown in basic or neutral soil, the flower is pink. So the information, the genetic information in the plants can be exactly the same, but when you change the pH of their soil, it can change the flower color you see. Another good example is with the snowshoe hare. In the wintertime, his fur is white. But as it becomes summertime, having a white rabbit really isn't going to work out very good. It's more susceptible to predation and who knows what else. So its fur actually starts to become a brownish or tannish color. And then what happens as winter approaches? That brownish or tannish color starts to change back to the white again. So in the wintertime, now he's got the protection from the white fur. And it keeps changing back and forth. The environment, the temperature, is affecting the coat color of that rabbit. And the other one that it's got here is light. Well, light doesn't maybe affect ours too much, but with plants it certainly does. If you take the light away, the chlorophyll no longer is expressed as much inside the plant. How do I know that? Lay something on your lawn. And if it lays on the lawn for too long, when you go back and lift it up, what happened to the grass underneath? It's turned that white or yellow color underneath that. Why did that happen? Because you took the light away, and for that chlorophyll to be expressed within the plant, it requires the light to make that happen. So the environment can actually have an effect on how the genes are expressed. They can have an effect on the DNA that's there. Now, does it actually change the DNA? Does it change the sequence of bases? No, not at all. All it does is affect the expression of that DNA in your outward appearance, in your phenotype. Your genotype, the genes that you have, the big A's or little a's or whatever's in there, that doesn't change, but the way that we show that may change. That takes us through and finishes up our introduction to genetics. Thank you for watching, and I'll see you next time.